glycolysis pathway, which is concentrated. Sorry, since my laptop is not working with the Google Meet, I have to teach you via another device. That's why I cannot attach my mouse, and I have to show you everything with the with this pencil on my slides. Okay, from the next session, so hopefully it will be in person and we will not have this kind of problem. Okay, so if you look at here, so you see in this slide glycolysis from here, glucose 6-phosphate, okay, here. So the point of this slide is to show that the glycolysis pathway could be replenished from different sites. What does it mean? It means that, for example, we have sucrose here. Sucrose by sucrase produce glucose and fructose. So this fructose enter glycolysis pathway from this point, because the glycolysis by glucose, it starts from changing glucose to glucose 6-phosphate. But when we have fructose, fructose change to fructose 6-phosphate and it enters from here. And if we have lactose, you know that lactose is the sugar in the milk. Okay, so the lactose on the function of the lactase, it changed to the glucose and to the galactose here. The glucose and here the galactose. Okay, you see galactose could be changed to glucose and it enters glycolysis as well. So the message of this slide is that glycolysis, it does not need only glucose. It could be performed by entrance of the galactose or fructose as well. Regarding the fructose, fructose change to fructose 6-phosphate and continue glycolysis, whereas galactose should be changed at the beginning to glucose, and as the glucose, it enters the glycolysis pathway. So in the second slides of today, I want to talk about the fructose metabolism in order to see how the fructose could be metabolized in our body. For example, if you eat honey, in the honey we have sucrose, and sucrose could be digested by sucrase on the epithelial cell of our intestine. It changed to the fructose, and as you saw in the previous slide, to the fructose and to the glucose. Okay, so let's see how the fructose could be metabolized in our body. If you look at fructose here, okay, we start from here. You see fructose here, and then fructose could be changed to either fructose 6-phosphate or fructose 1-phosphate. So fructose 1-phosphate by the function of enzyme known as fructokinase. If it changed to fructose 6-phosphate by the function of enzyme known as hexokinase. Okay, so we can change fructose to fructose 1-phosphate or to fructose 6-phosphate. It depends in which tissue of the body the fructose is metabolizing. For example, if it is in the liver, the fructokinase phosphorylate fructose to fructose 1-phosphate. But if we talk about the adipose tissue or fat tissue, fructose is metabolized to fructose 6-phosphate by hexokinase. So the first reaction is phosphorylation of fructose on the position of carbon 1 or position of the carbon 6. So if we continue here, you see that fructose 1-phosphate by aldolase B breaks to dihydroxyacetone phosphate and diglyceraldehyde. Fructose has 6 carbon. It breaks to 2 molecules of 3 carbons, glyceraldehyde and dihydroxyacetone phosphate. So what happens next? The diglyceraldehyde by ATP and tyroskinase change to glyceraldehyde 3-phosphate. Okay. How about dihydroxyacetone phosphate? Either it is used for fatty acid esterification. I will explain it later. What does it mean? Or it is changed to glycer 3-phosphate as well. How we can change dihydroxyacetone phosphate to glyceraldehyde 3-phosphate? By the enzyme phosphotheriose isomerase. Then glycer 3-phosphate, it changed to 2-phosphoglycerate and pyruvate. I mean, from here, from this point, we are in the glycolysis pathway, because I told you fructose can enter glycolysis. But from this part, it enters by glyceraldehyde 3-phosphate. But if we talk about this pathway, it enters the glycolysis by fructose 6-phosphate. Okay, let me see if I can use another color to show it better. Okay, so how about the hexokinase? By the hexokinase, we change fructose to fructose 6-phosphate. Then the enzyme phosphofructokinase, this enzyme, by using ATP, it changed fructose 6-phosphate to fructose 1,6-bisphosphate. You should remember we had fructose 1,6-bisphosphate in the glycolysis. Then the rest of the reaction here, these are all the same as glycolysis. We produce pyruvate, then pyruvate could be used for the fatty acid synthesis. Okay, that's why, since the pyruvate could be used for fatty acid synthesis, I will explain you in the lipid metabolism, because you will see there pyruvate could be changed to acetyl-CoA, and acetyl-CoA is the precursor of fatty acid synthesis. The more pyruvate we have in our body, the more fat could be produced as well. That's why they say the people who use too much fructose, and since the fructose could be metabolized more quickly compared to the glucose in the glycolysis pathway, we reach to pyruvate rapidly compared to glucose in the glycolysis, and then we will have rapidly acetyl-CoA, and then this acetyl-CoA could be used to produce fatty acid. And if you have heard the disease known as fatty liver disease, so the more consumption of the fructose due to this metabolic pathway could be used, it could increase the incidence of fatty liver disease, because it is metabolized in the liver and it can produce acetyl-CoA, it can produce fat. These fat accumulate in the liver, it causes fatty liver disease. So these two pathways which I explained for you regarding the fructose metabolism, please keep in mind. The important part is fructokinase and hexokinase and the product and the rest of things which I explained to you. So let's have a look at the top of this diagram. Okay. So here you see we have glycogen. The glycogen, upon degradation, produce for us glucose 6-phosphate. Okay. So the glucose 6-phosphate by phosphohexoisomerase could be changed to fructose 6-phosphate. The rest of metabolic pathway is glycolysis. If we look at here, so the glucose 6-phosphate could be changed to glucose by which enzyme? Glucose 6-phosphatase, because it removes the phosphate from carbon number 6. Then the glucose by the enzyme known as aldose reductase the enzyme known as aldose reductase. The glucose, by using NADPH, the electron from NADPH, it changed to the sorbitol. Okay, and sorbitol could be changed to fructose. It means that from glucose in our body, we can produce fructose as well by these metabolic pathways. Yes. Sorry? There's no presentation? Okay, let me share it again. How about now? You do not see the slides? Okay. Let me see why. How about now? Do you see the slides? Okay. And my slide disappeared in which reactions? I was explaining which reaction. Do you understand what I mean? I mean, you heard my explanation on the slide up to which reaction?
We speak about phosphorated 6-phosphate. Fructose 6-phosphate.
Fructose. It means, let me erase this part to see. So
From
you
glucose
heard,
yes.
you heard this part, right? This part you
Yes.
heard. I
Yes.
assume you didn't hear the top. Okay,
We start
so,
from the top and disconnect.
Okay, so now I will explain it again, no worry. Okay, so I told you that in the top we have glycogen, okay? Glycogen, upon degradation, produce for us glucose 6-phosphate, okay? Then this glucose 6-phosphate, either it could be changed to fructose 6-phosphate. You remember this was the reaction in the glycolysis by the enzyme phosphohexose isomerase. Or glucose 6-phosphate changed to glucose by glucose 6-phosphatase. Do you remember this enzyme? We had it in the gluconeogenesis as well. So the first reaction of glycolysis, hexokinase, was irreversible, and it could be returned back by the enzyme glucose 6-phosphatase, in which we change glucose 6-phosphate to glucose. So we produced glucose, and glucose by the enzyme known as aldose reductase changed to sorbitol. Here we need the electron from NADPH because the enzyme is reductase, meaning that we need to reduce glucose to another product. So then sorbitol by enzyme sorbitol dehydrogenase changed to fructose. NAD reduced to NADH, because when we want to change sorbitol to fructose, we need to oxidize sorbitol to fructose. So in this pathway, you could see we can produce fructose from glucose in our body, okay? Because here it's mentioned diet, but we do not need always to have fructose from diet. It could be produced in some specific cells in our body. Which cells? For instance, spermatozoa in men, because the spermatozoa needs fructose, and fructose could be produced in the spermatozoa from this pathway at the top, which I explained you, the change of sorbitol to fructose. And if you look at here, so the sorbitol to fructose could be reversed as well. The fructose could be changed to sorbitol as well. Sorbitol could be changed to glucose as well. So Another point regarding the sorbitol is that in any cells we have sorbitol. Sorbitol is able of making osmotic pressure. Since it increases osmotic pressure, therefore this osmotic pressure leads to accumulation of water in that cell. Okay, and in the diabetic patient, since the glucose is high in the blood of diabetic patient, so those cells which can take too much glucose in the cell, and if they have this enzyme, aldose reductase, they can change glucose to sorbitol, and the sorbitol can damage that cell, because I told you that sorbitol can make osmotic pressure, and also it can lead to accumulation of the water in that cell. So some people believe that the problem in the eye of diabetic patient is due to activation of this mechanism, this pathway in the eye cell. So the high level of the glucose in the eye cell change to sorbitol, and sorbitol, based on the mechanism which I explained to you, can cause problem in this cell. Okay, so regarding the pathway fructose metabolism, which I explained you, two disorder has been reported. One of them is essential fructosuria. So it is due to the deficiency of fructokinase. Where did we have fructokinase? Fructokinase was here. Okay, so fructokinase was responsible for changing fructose to fructose 1 phosphate. If this enzyme is absent, no more conversion of the fructose to fructose 1 phosphate occur, and the person will excrete fructose in the urine. The person will show fructosuria, meaning that the fructose appear in the urine. And another disease is hereditary fructose intolerance. So in this disease, the absent enzyme or deficient enzyme is aldolase B, this enzyme, okay? If the aldolase B is absent, so fructose could be changed to fructose 1 phosphate, but the fructose 1 phosphate could not be broken to glyceraldehyde and dehydroxyacetone. We will have accumulation of the fructose 1 phosphate. This patient will suffer from the hypoglycemia because the accumulation of fructose 1 phosphate interfere with the glycogenolysis in the body. Therefore, since the glycogen could not be broken down, therefore the level of the glucose become low. The person will suffer from hypoglycemia. So this was the story of the fructose metabolism. Now I want to talk about the galactose metabolism. Okay, so regarding the galactose metabolism, so if you look at here, so you see that we start with the galactose here. Okay, we start with the galactose. The first enzyme is galactokinase. This enzyme using ATP can phosphorylate galactose to galactose 1 phosphate. Then galactose 1 phosphate, if you remember in the first slide which I explained to you, I talked that fructose and galactose can interglycolysis, but fructose interglycolysis upon its phosphorylation, but galactose should be changed to glucose. It changed to the glucose, then it interglycolysis. Therefore, here we should see how we can change galactose to glucose. So the first reaction, galactose to, we have conversion of the galactose to galactose 1 phosphate. Then we have one enzyme known as galactose 1-phosphate uridyl transferase. It means that this enzyme can transfer UDP to the galactose. Okay, so here we have galactose 1-phosphate, here we have UDP glucose. Okay, so what happens here, this enzyme can transfer this UDP to galactose, therefore it changed to UDP galactose, and glucose changed to glucose 1-phosphate. Okay, so here we could change galactose 1-phosphate to UDP galactose by accepting UDP from glucose, and glucose changed to the glucose 1-phosphate. So then in the next reaction, the enzyme uridine diphosphogalaktose 4 epimerase, you should have learned that galactose and glucose, these are epimer of each other, 4 epimer. Okay, so the enzyme uridine diphosphogalaktose 4 epimerase, it can change galactose to glucose. Okay, so this change galactose to UDP glucose, then we have reached to the glucose. So if we look at these parts, when we produce UDP glucose from we produce UDP, oh my God, we produced UDP glucose from galactose. Now it could be used in the other pathway. For example, in the glycogenesis, the UDP glucose could be used in order to making glycogen. You remember when we made glycogen, we need to have activated form of the glucose, which was UDP glucose. Here we produce UDP glucose from galactose. It could be used in the glycogenesis. So we produce glycogen, then if we need, the glycogen could be broken down in order to produce for us glucose, which I explain you in the glycogenolysis. So regarding the disorder reported in the galactose metabolism, so it has been reported one disorder known as galactosemia, so which is caused by inherited defects of galactokinase, I mean this enzyme. Upon defect in this enzyme, the disorder is known as galactosemia. So the deficiency is also reported in the second enzyme, uridyl transferase, or in the 4 epimerase. Though deficiency of uridyl transferase is best known, I mean this one is best known. And if there is deficiency of galactokinase, so we will have the reduction of galactose to galactitol, because galactose could not go through this direction. So the galactose reduced to galactitol, and it is believed that the produced galactitol result in the process or in the phenomena which is known as cataract. This is also the problem in the eyes. The last pathway for the carbohydrate metabolism is glucuronic acid pathway, which I explained to you in the previous session. We said that this is another oxidation pathway for the glucose. It is present only in the liver, and it does not produce any ATP, the same as pentose phosphate, which we didn't get any ATP. But it's important because the glucuronate produced in this pathway, it's necessary for the detoxification of some toxins in our body. And it can convert the glucose to glucuronic acid, also to ascorbic acid, not in us, but in for example, because it's mentioned that this conversion, the conversion of the glucose to ascorbic acid in the glucuronic acid pathway, it does not occur in the human beings. And you know that the ascorbate or ascorbic acid, which is vitamin C, it's considered as vitamin for us. We cannot produce it in our body. Okay, so our lecture on the carbohydrate metabolism is done. If there is any question on these 3-4 slides which I just explained to you, please feel free to ask me. Otherwise, I will switch to the lipid metabolism, the next lecture. Any question, guys? Okay, seems there is no question. Okay, so the next lecture, in this session and next session, probably the next session as well, we will talk on the lipid metabolism. Regarding the lipid metabolism, we are going to talk about the digestion and absorption of the lipid, how we can oxidize fatty acid or fatty acid oxidation, how we can synthesize fatty acid in our body, fatty acid biosynthesis. We introduce you ketone bodies, the metabolism of ketone bodies. We will talk about the metabolism of the cholesterol and metabolism of the lipoprotein. Okay, guys, in this slide, so you see that via the diet, the lipid is entered in the mouth and in our digestive tract. So you see that it is composed of CE, which is cholesterol ester. So the picture is shown on the right side of this slide. CE is cholesterol ester. And if we want to produce cholesterol from cholesterol ester, the enzyme cholesterol esterase can remove the bound fatty acid by the aid of water in order to make cholesterol. So it's also composed of PL or phospholipid, TAG or triacylglycerol. So the lipid remain unchanged in our mouth. If you remember, for the carbohydrate metabolism or digestion, we said that alpha amylase, the salivary alpha amylase, it starts to break 1-4 linkage in the mouth. But you see that the digestion of, although the digestion of carbohydrate occur in the mouth, but the lipid remain unchanged in the mouth. So in the stomach and in the small intestine, so you see that these lipids are moving, they reach to the small intestine, and you see that we will have enzyme from the pancreas, the lipase, which can start to digest these lipids. And in the next slide, I will explain you better, just to have a whole picture, a schematic picture. I will continue here, then we go to the next slide. So the bile salts, it emulsify, and pancreatic enzyme, they degrade dietary lipid. And the primary products of this degradation or digestion is free fatty acid, because, for example, here, the cholesterol ester, it results to production of fatty acid and cholesterol. Therefore, here we say we will have free fatty acid. 2-monoacylglycerol and cholesterol. Okay, cholesterol, you saw it here. What does 2-monoacylglycerol means? If you look at here, we have triacylglycerol, and the enzyme pancreatic lipase, by the water, it removed 2 fatty acids, and the only remaining fatty acid is on the carbon number 2. Therefore, it is shown as 2-monoacylglycerol. So we have the phospholipid here, PL. You see the lipase by the water remove fatty acid, and it can, for example, change. For example, choline is one of the phospholipid. By the function of lipase, it changed to glycerophosphorylcholine. So after we produce these primary products, these are absorbed in the epithelial cell, and These are re-esterified again, and they are packaged in the lipoprotein known as chylomicron. It enters in the lymph and into the blood. Regarding the chylomicron, which is a kind of lipoprotein, no worry, I will explain you in this session of lipoprotein metabolism. Okay, so this is how the lipid could be digested and absorbed, but here I want to talk about the role of hormones in the control of the lipid digestion in the small intestine. Okay, so at the top you see a stomach, so you see the pancreas in brown, you see gallbladder in green, and you see gut endocrine cells in yellow. Okay, so here you see that upon the entrance of the dietary lipid, let me show you with this. Okay, upon entrance of the dietary lipid to the small intestine, okay, we will get the positive feedback on the gut endocrine cells. So the gut endocrine cells, it will respond to this feedback by release of the cholecystokinin in the blood and secretin in the blood. Okay, the cholecystokinin result in, it will have negative feedback on stomach because it wants to make the stomach understand it should not, for example, release the other, for example, food or the other things in the stomach into the intestine. And the secretin in the blood, it has positive effect on the pancreas, and it results in the secretion of bicarbonate into the small intestine. If you look at cholecystokinin, it has also positive effect on the pancreas and gallbladder as well. So on pancreas, by its positive effect, release pancreatic enzyme, and gallbladder release bile. Therefore, all of these things, pancreatic enzyme and bicarbonate and the bile, they result in degradation of dietary lipid. Bicarbonate is important because it can provide the favorite pH for pancreatic enzyme in order to degrade the lipids, because you know that in the stomach we have acidic pH, and since these are released in the intestine, we need to provide the alkaline condition for this pancreatic enzyme. And as you saw in the previous slide, the pancreatic enzyme, which are lipase, these lipase are involved in degrading the lipids. For example, here you saw that pancreatic enzyme, it can change 3-acylglycerol to 2-monoacylglycerol, and the lipase changed choline to glycerophosphoryl choline. Okay, so this was the digestion and absorption of the lipid. Okay, so now the lipid is absorbed in our body. What does these lipid include? So when I say lipid, it means that the lipid include fatty acid, it includes the lipid soluble vitamin, and it includes the cholesterol, it includes, for example, everything, okay, phospholipid, everything. So then these are distributed to our cells. These are absorbed from intestine. Now it is in the circulation. It is delivered to the liver, and the liver is distributing this lipid. Okay, so I said in the liver, but when I talk about the lipoprotein metabolism, you will understand better that when these lipids are packaged in the chylomicron, because here I told you that the absorbed lipid is packaged in the chylomicron. So the chylomicron is passing, it's moving in the circulation, and in the circulation it delivers the, for example, fatty acid to the tissue which needs fatty acid. So now you imagine that the fatty acid in the chylomicron is delivered to the, for example, adipose tissue, to the fat tissue. And the fat tissue wants to use this fatty acid, which came from the diet. So how does it use? Let's say that the fat tissue needs to have energy from fatty acid. Therefore, it needs to oxidize fatty acid. It needs to oxidize fatty acid. We have different way of fatty acid oxidation. For example, we have omega oxidation, we have alpha oxidation, we have beta oxidation. For you, I am going to talk only about the beta oxidation. Okay, so what does beta oxidation means? If you remember the structure of the fatty acid, which is explained to you previous semester, so this is the fatty acid, but it is fatty acyl-CoA. Why fatty acyl-CoA? Because the coenzyme A is bound to this fatty acid. If this part we had COOH, we call it fatty acid. But now I call it fatty acyl-CoA because one coenzyme A is bound to fatty acid. So it should have been told you that the carbon is, which is the neighbor of this carbon in fatty acid, we call it alpha. This one we call it beta. Okay. So if we, let me have it. If we break this linkage from here, I mean between alpha and beta, okay, we call it beta oxidation because we have oxidized fatty acid and we have broken fatty acid from the bond which was between carbon alpha and carbon beta. When we oxidize it in beta oxidation, so this is released, and this is known as acetyl-CoA because it has two carbons with the coenzyme A bound to it. And if you consider this acetyl-CoA, this fatty acid-CoA has, for example, 16 carbon. Okay, this acetyl-CoA has 16 carbon. If beta oxidation occurs and we remove one acetyl-CoA, how many carbon it will have? 14 carbon, because 2 carbon is already lost via beta oxidation. This is the first round of beta oxidation. So if we continue the second round of beta oxidation, then we will have 12 carbon. The remaining part of fatty acid is 12 carbon. We continue it, we will have 10 carbon because in each round we are removing one acetyl-CoA from our fatty acid. Okay, so this is what I needed to explain you about the beta oxidation in order to understand why this kind of oxidation is known as beta oxidation. Okay, and it's mentioned that the beta oxidation of fatty acid, it involves successive cleavage with release of acetyl-CoA. So the oxidation of fatty acid is a little bit complicated because when I say that, for example, oxidation of fatty acid involves cleavage of fatty acid by releasing acetyl-CoA, it's not true about all the fatty acid, because if the fatty acid is odd number, okay, if it is odd number, for example, let's say that it has, for example, 17 carbon. Okay, if it has 70 carbon, we do not produce acetyl CoA. I mean, we do not only produce acetyl CoA, because we will produce several acetyl CoA. We will also produce one propionyl CoA. Propionyl CoA has 3 carbon. But if the fatty acid has, for example, 16 carbon, 14 carbon, 12 carbon, so all the product of beta oxidation is acetyl CoA. But for the fatty acid, which include, for example, 13 carbon, 15 carbon, 17 carbon, 19 carbon, we do not have only acetyl CoA as the product of the fatty acid oxidation. We always will have acetyl CoA plus propionyl CoA. Therefore, when we talk about the fatty acid oxidation, it could be complicated. So meaning that we should keep in mind whether the acetyl CoA is, for example, odd or not. If the acetyl CoA has double bond or not, because you know that we have saturated fatty acid, we have unsaturated fatty acid. So, but for you, I am going to talk only about the saturated fatty acids, okay? And the example which I give you is the fatty acid with 16 carbon. Therefore, the product of beta oxidation of this fatty acid, 16 carbon, which is known as palmitate, is acetyl CoA. We will not get any propionyl CoA. Okay, so you understood what does beta oxidation of fatty acid means. Now let's say how does the beta oxidation occur in the cell. So the first important thing is that to know where is the location of beta oxidation. So for example, regarding glycolysis, I told you glycolysis occur in the cytoplasm of cell. But beta oxidation of fatty acid occurs in the matrix of mitochondria. Therefore, we need to reach fatty acid inside the mitochondria, in the matrix of mitochondria. But there is one problem. The problem is that the inner membrane of the mitochondria is not permeable to all of the fatty acid. So if the fatty acid has short lengths, if it has short lengths, therefore, or it could be passed through the inner membrane. But by increasing its length, it cannot pass through. For example, the fatty acid with 16 carbon, it's impossible. Therefore, we need to have a system in order to pass the fatty acid from the inner membrane of the mitochondria into the matrix of mitochondria. So this system is using carnitine and different enzymes. Therefore, I need to explain you how this system works. First of all, we should we should reach the fatty acid inside the matrix, and then we will talk about the several reactions which are involved in the beta oxidation. And if you look at here, it's mentioned that long-chain fatty acid penetrate the inner mitochondrial membrane as carnitine derivative. Okay, guys, so let's look at here. So here you see the mitochondria. Let me show you by the color. So this is the outer membrane of the mitochondria. It's bilayer. This is the inner membrane of the mitochondria. Again, it's bilayer. And here is the matrix of mitochondria. Here is the space between outer membrane and inner membrane. So the FFA represent free fatty acids. Okay. the free fatty acid has passed through the plasma membrane of the cell. It has reached the cytoplasm of the cells. Now it wants to pass through the two membrane of the mitochondria in order to reach the matrix of mitochondria. So the FFA, first of all, should become activated. Do you remember regarding glycogen synthesis? I told you that first of all, we need to activate glucose to UDP glucose. Here we need to activate fatty acid to acyl-CoA. What does acyl-CoA means? When I say acyl-CoA, it means that I have added coenzyme A to fatty acid. Okay, so I said acyl because I have not given you any information on the chain of the fatty acid. I didn't tell you how many carbon we have in this fatty acid. That's why I say acyl-CoA. So when we want to change FFA to acyl-CoA, we use 2 molecules of ATP. We need to have energy. ATP is changed to AMP. This is equal to 2 molecules of ATP. We had coenzyme A as well. The enzyme responsible for changing FFA to acyl-CoA is known as acyl-CoA synthetase, located in outer mitochondria membrane. Then acyl-CoA pass through outer mitochondrial membrane, and here you see we have one enzyme known as carnitine palmitoyl transferase 1, abbreviated as CPT1. The acyl-CoA, by the function of CPT1, release its CoA and it binds to carnitine. Then we produce acyl-carnitine. What did we do so far? So far, we could change FFA to acyl-CoA. Acyl-CoA entered the space between two membranes of the mitochondria. Here, CPT1 changed acyl-CoA to acyl-carnitine. It removed the CoA part and added carnitine to acyl part. So acyl-carnitine is produced. Why did we need to change fatty acid to acyl-carnitine? Because in the inner membrane of the mitochondria, we have this guy, carnitine acyl-carnitine translocase, which can pass acyl-carnitine to matrix of mitochondria, not the acyl-CoA, not the fatty acid. Therefore, we didn't have any other choice than changing free fatty acid to acyl-carnitine. Okay, acyl-carnitine passed through inner membrane of the mitochondria by this translocase, and here we are in the matrix of mitochondria, and now we should change acyl-carnitine to acyl-CoA again, because in the beta oxidation we start with acyl-CoA, not with acyl-carnitine. So this acyl-carnitine by another enzyme here known as CPT2, carnitine palmitoyl transferase 2. In the matrix, it releases the carnitine from acyl-carnitine and removes the carnitine from acyl-carnitine, and it adds coenzyme A to the acyl part. So the carnitine is released, carnitine goes back to the space between outer and inner membrane of the mitochondria, and the CoA is bound to the acyl part and produced for us acyl-CoA. So this acyl-CoA should enter beta oxidation pathway, because the beta oxidation is inside the matrix of the mitochondria. Any question, guys, so far? Do you have any question? No? So up to here, we could deliver our fatty acid to the matrix of mitochondria, where all the enzyme required for oxidation of fatty acid is present. So the enzyme which are which can oxidize the fatty acid in the matrix, so are shown here. Okay, so this is for example the enzyme which are all together. Okay, so you see we have different enzyme. I will explain this enzyme when I'm talking about the reaction. So this is the overview of beta oxidation of fatty acid. So the fatty acid in the first reaction, we said it should be changed to acyl-CoA by using 2 ATP, and then it could change from the cytoplasm to the matrix of the mitochondria. And if I want to give you another overview of beta oxidation, here you see that, for example, we have palmitoyl-CoA. So the palmitoyl-CoA with 16 carbon, the beta oxidation, you see, it breaks the linkage between carbon alpha beta. So one acetyl-CoA is removed, then it continues by removal of another acetyl-CoA. We will reach to 8 acetyl-CoA, because I said palmitoyl-CoA has 16 carbon. So upon this cleavage, we reach to 8 acetyl-CoA. Each acetyl-CoA has 2 carbon, therefore 8 multiplied by 2 carbon, we will reach to 16 carbon. Okay, so this is the reactions involved in beta oxidation of fatty acid. So you remember that I told you the acyl-CoA, how it should be entered to the matrix of mitochondria. Now we are in the matrix of mitochondria, and acyl-CoA is ready for beta oxidation. So the first reaction, the enzyme known as acyl-CoA dehydrogenase, by using FAD, oxidize acyl-CoA to this guy. You see, between carbon number 2 and number 3, we will have double bond upon this oxidation. So we produce delta 2 trans-enoyl-CoA. In the next reaction, one hydratase, which is known as delta 2 enoyl-CoA hydratase, by the water, by the molecule of the water, it produces for us 3-hydroxy acyl-CoA. Then the next enzyme, L3-hydroxyacyl-CoA dehydrogenase, produce 3-keto acyl-CoA. Okay, if I want to show you here, so delta enoyl-CoA hydratase, you saw we had double bond between carbon 2 and 3, and then this dehydrogenase, it could oxidize this guy, reduce NAD to NADH. And by this oxidation, you see it produced this CO on carbon number 3. So the guy is known as 3-keto acyl-CoA, because you see that on the carbon number 3, we have a carbonyl group instead of having a hydroxyl group. Okay, so then in the fifth reaction, the enzyme thiolase perform on the 3-keto acyl-CoA. So you saw that 3-keto acyl-CoA is produced here. Okay, so by the entrance of coenzyme 8, the thiolase, it can remove this part, the coenzyme added to the this part. It can remove the 2 carbon at the end and produce for us acetyl-CoA. Acetyl-CoA enter Krebs cycle. The remaining part goes back to the this reaction because it wants to lose another 2 carbon, another acetyl-CoA. We said that if it has 16 carbon, we need to perform this metabolic pathway 7 times until we oxidize until we oxidize whole of 6 carbon fatty acid. Okay, so guys, is there any question? Is there any question here? Okay, so you could understand how beta oxidation of fatty acid occur, and here you saw that upon oxidation of fatty acid, for example, in one round. So if we want to take one acetyl-CoA, we remove one acetyl-CoA, one FADH2 is produced here, one FADH2, one NADH is produced here as well, meaning that when we remove one acetyl-CoA from fatty acid, we will have one FADH2 and one NADH. Therefore, if I want to calculate the energy which I get from oxidation of C16 fatty acid, here you see that I have 16 carbon fatty acid. Now I want to understand how many ATP do I get from beta oxidation of 16 carbon fatty acid. I said that for complete oxidation of this fatty acid, I need to perform this pathway 7 times. 7x means that I should do this pathway 7 times in order to produce 8 acetyl-CoA, because if I want to oxidize all of 6 carbon, I should do 7 times, okay, to reach to 8 acetyl-CoA. 8 acetyl-CoA is equal to 16 carbon. Why didn't I say 8 cycle 8 times? Because at the end I will have 4 carbon and the thiolase produce 2 acetyl-CoA at the same time. Okay, so if you look at this table, so for the 16 carbon fatty acid, I produce how many acetyl-CoA? 8 acetyl-CoA and 8 acetyl-CoA, okay? And if you remember, in the, why my marker does not work? Okay, 8 acetyl-CoA. And if you remember Krebs cycle, we said from each acetyl-CoA we produce 10 ATP. Therefore, the 8 acetyl-CoA produce for us 80 ATP, 80 molecule of the ATP. So since we said the, oh my God, what is this ruler here? Okay, so we said for each acetyl-CoA production from fatty acid beta oxidation, we produce one FADH2, one NADH. Since we need to repeat it 7 times, we say 7 ATP, 7 FADH2 and 7 NADH should be produced upon complete oxidation of 16 carbon fatty acid. 7 FADH2 multiplied 1.5 is equal to 10.5. 7 NADH multiplied by 2.5 is equal to 17.5. If we sum all, we reach to 108. 108. And if you remember, we said that for activation of the fatty acid, we used 2 ATP. Okay. Therefore, if I remove these 2 ATP from 180, I will reach to 106. Therefore, if I oxidize C16 fatty acid, okay, in beta oxidation, I will reach 106 ATP. Did you understand how I have calculated 106 ATP? I don't really know if you are there or not. Okay. Okay, regarding the beta oxidation of the fatty acid, so this was about the 16 carbon fatty acid. Imagine that we have a, for example, very long fatty acid, very long chain fatty acid, for example, 26 carbon, 24 carbon. So these very long chain fatty acids, first of all, should be oxidized in the organelle known as peroxisome. So first of all, they oxidize in the peroxisome, and then they will be oxidized in the, the oxidation will be continued in the matrix of mitochondria. Okay, I think it's enough for today. So the rest of the slide I'm going to talk about in our in-person session, hopefully next week. Is there any question, guys? Please feel free to ask me.